All right, let's jump into it. Now we have some people here. So hello, everybody, and thank you for joining today for our live virtual demo of the OsmoTECH XT single sample micro-osmometer and the OsmoTECH PRO multi-sample micro-osmometer. My name is Eric Chanel, and I am the Global Product Manager for our OsmoTECH portfolio. And with me today, I have Ben Douglas, our North American Field Application Scientist. I'm going to run through a quick few slides before passing over to Ben for the live demo. Here we go. So just a quick bit of housekeeping. We'd love to hear your questions. So please feel free to enter them either in the chat or put them in the Q&A session. Either one I'll be monitoring throughout. And so I'll either be answering them during the conversation, passing them to Ben, or there'll actually be some extra time at the end as well, most likely. So lots of different ways to get your questions in here. So to kick us off, I'm going to give you a quick introduction to Nova Biomedical. So in the Advanced Instruments brand, we are the 70-year global leader in osmolality testing, experts in diagnostic and critical care, cell culture analysis, cell line development technology, and liquid handling quality assurance. We support customers globally. We have installs across 90 countries throughout North America, EMEA, and the APAC region, focusing on developing technologies that streamline your workflow and really build some confidence in your results. So we currently offer 5 different portfolios that serve our customers in the bioprocessing market. Today, we're going to focus on the Advanced Instruments OsmoTECH portfolio of osmometers. The OsmoTECH is the only osmometer portfolio that's designed for customers in the bioprocessing workflows. They're optimized for testing of biological samples, such as cell culture media, buffers, and aqueous drug products. The other 4 portfolios are the Nova Bioprofile portfolio, the Flex 2 cell culture analyzer, and the Fast CDV cell viability analyzer. The Celentum portfolio, which is for single cell cloning, assurance and cell characterization, and cell line development instruments workflows. And the Artel instruments for pipette and automated liquid handling calibration. And lastly, the Enoximat anaerobic jar system for bacterial cultivation. All these product portfolios are supported by our own dedicated Nova Biomedical trained FSEs, so field service engineers, and we have service contract options available to help you maintain this equipment should anything come up, so you don't have any disruption to your workflow. Specifically for the OsmoTECH portfolio, we offer qualification and validation services to ensure regulatory compliance and help you get your instrument up and running quicker. Now, just to focus in on osmolality testing here, let's discuss some of the applications of osmolality testing in biological development and the manufacturing processes. Starting with cell line development and throughout cell culture scale-up, osmolality can be used to confirm the concentration of media, feeds, and can help you gain insight into optimizing the scale-up steps. In downstream purification workflows, osmolality is used to confirm buffer concentrations. It can help ensure the buffer exchange and can measure concentration of the drug substance. And finally, in formulation, all the way through development and through fill and finish, osmolality is a critical quality attribute that provides valuable information about the identity, safety, purity, and stability of the drug. I mean, for these injectable drug products, the osmolality solution must be within that physiological range, or it must be close to the osmolality of blood, since anything higher or lower can cause pain at the injection site, which can cause problems with patient compliance. Now, how do you pick the best osmometer? Well, for when you're looking for the new one for your lab, there's some features you probably want to consider. And these are in no particular order here, but is it simple? Is the instrument accurate? Is it reliable? And how much volume does it take? Volume's quite precious. The sample's quite precious, so you don't want to waste much of it. How long does the test take? Your time's valuable. How do we record the data that we generate? And how do we save it? And will the instrument fit into your workflow in terms of regulatory compliance? So the OsmoTECH osmometers are built with your needs in mind. All those needs I was just talking about, and when you're making those considerations, that's what we built it towards to fit all of that here. The testing process, it's fast and simple, easy to integrate into any workflow or in your different types of throughput. We have flexible data connectivity options, strong data integrity features, and these features are adjustable to your lab's specific needs. They are flexible for R&D, for process development, or for manufacturing settings. And we'll show you this throughout where we'll have it set on some less restrictive or more restrictive throughout the process so you can see how it all looks. And with the OsmoTECH XT specifically, you can test samples up to 4,000 milliosmoles and improve testing performance with those viscous or highly concentrated samples that you'll see other instruments struggle with. All of the OsmoTECH portfolio micro-osmometers currently support the different regional pharmacopoeias, such as EP, USP, JP, KP, and more. Today, we're going to focus on two instruments. We're going to focus on the OsmoTECH XT single sampler and the OsmoTECH Pro multi-sampler. So to compare these instruments, starting with the XT, you have the operating range that I mentioned of 0 to 4,000 milliosmoles. You have a test time of roughly 130 seconds in the low range, about 180 in the high range, and Ben will get into what that difference is. You have one-step direct testing method and a really, really small volume requirement of just 20 microliters. The OsmoTECH Pro is your multi-sample. A little more restrictive of a range of 0 to 2,000 milliosmol, but a quicker test time, 90 seconds, using a sample cup, and again, small volume requirement of 30 microliters. And we'll go through both of these instruments in detail. And with that, let's get to the fun stuff. So I'll give a quick pause to see if there's any questions. If not, I will exit from this and move over to Ben. Bear with me one moment while I do that.
Sorry, everyone, for that. So again, sorry for that. My name is Ben Duckless. I'm the field application scientist here for North America for the Osmo Bio portfolio. As Eric said, we're going to be spending some time today with the OsmoTECH XT and the OsmoTECH Pro. We're going to be going over some of the key pieces of the hardware and software for both instruments. We're going to be spending a little bit more time with the OsmoTECH XT today as far as the dive into the software goes, because they're virtually the same across both instruments. So we're not going to replicate our efforts doing both deep dives into the software. We'll start with the XT. We'll move to the OsmoTECH Pro. And then, as Eric said, we'll factor in some questions throughout the presentation, and then we'll answer the bulk in real time in the chat box with that Eric's monitoring. And then we'll also take some time towards the end of the chat to answer any remaining questions that we have. All right. And with that, we will dive into the OsmoTECH XT. And we'll just switch over to the camera real quick. There we are. Okay, so here's our OsmoTECH XT. So like I said, I'm going to go over some basics of the hardware and consumables before diving into the user interface and the software. So as Eric mentioned, this is our purpose-built osmometer for the bioprocessing development teams in the biotech industry. Just to reiterate, it has a working range of 0 to 4,000 milliosmoles that's split into two ranges, low and high. So our low range incorporates 0 to 1,500 milliosmoles, and our high range incorporates 1,500 to 4,000 milliosmoles. It's a single sample direct read. So we are going to be utilizing our single sample sampler right here. So this is pre-calibrated to pull that 20 microliter sample from either our reference standards or our unknowns. That's fed directly into the instruments utilizing a sample tip, which is housed in the consumables box up top here. So within this, we have 100 sample tips as well as 100 cleaning wands. And we'll dive into what these cleaning wands are used for as we run a sample. But to attach the tip, it's quite simple. Just fits right on top of the plunger right here. Snap it in place, and we're good to go. So this is where the actual read's going to be taking place. So we have an opening up top here where we will pull up our sample or our reference standards. We'll load it directly into the system, and then the test is performed directly in the sample tip. It's single use, so once we're done testing, we'll pop it off, throw it in the trash, and then we'll use another one to run an additional sample, or as many samples as we have throughout the process. Let's close this up real quick. Directly next to it, we have our sample cradle. So this is where our sampler is going to interact with the cooling chamber. So we're just going to slide this up once we're ready to run our sample. All right. And then directly behind the system here, we have a very simple, easy to use on-off button. It's the only button on the system. It doesn't require additional long, prolonged calibration times or warm-up times. It takes a few moments for the instrument to initialize once it's turned on. And then we're essentially ready to start testing as soon as that process is complete. And a little tough to see in the back here, we also have dedicated USB ports. It can be used to attach peripherals to the instrument, so a keyboard or a mouse. If you guys don't want to interact with the glove-friendly touchscreen up front here, you have the option to use those accessories, for increased ergonomics or if you guys prefer to do that rather than utilizing the touchscreen. We also have an Ethernet port here, so it's the OsmoTECH XT as well as the OsmoTECH Pro do have the abilities to interact with your local area network. If you guys utilize LIMS or LIS or shared repositories that you'd want to copy your data to from the systems, you have the ability to do that using the Ethernet port and local area connection. We also have a port in the back here to connect a dot matrix printer if you guys need to utilize printer-based reporting methods per your SOPs. But we tried to think of a few different ways for you guys to make your data management in the lab a little bit easier. The USB ports, can also double as a way to transfer data on and off the instrument should you guys need to as well. So you have the ability to export data directly to a USB drive from the XT and the OsmoTECH Pro, if that is part of your workflow that you'd want to utilize. All right. Moving to the front of the screen here, so we have our, as I mentioned, our glove-friendly touchscreen. It's very user-friendly and simple design and layout, which makes using the system and interacting with the system very easy on a day-to-day basis, especially if you guys are running quite a lot of samples in the lab. Very simple to use, and we'll get into this in just a moment. And the last piece that I want to touch on is our integrated barcode scanner right in the front here. So it's great, if you guys are running a lot of samples in the lab that have pretty lengthy sample names or sample ID tags. You guys want to utilize barcodes to keep track of those samples when you're running the osmolality test. It just makes life a little bit easier for you guys. But you do have the ability to include sample tags using free text with the touchpad or the keyboard should you guys want to do that. All right. Next, what I want to do is just do a quick round robin of the home screen here. So go over some of the various icons before diving into some of the main submenus to really kind of hammer home some of the nice features of the OsmoTECH XT and the OsmoTECH Pro that really make it compliant for the 21 CFR Part 11 as well as the EU Annex 11 regulation compliances that Eric mentioned earlier. All right, so we're just going to work clockwise starting from the top left here. So keeping in mind of the 21 CFR Part 11 compliance, we do have the ability to assign unique user IDs and passwords to the system. Right now, it's in its basic form, so anyone can kind of walk up and use it. But you do have the ability to set restrictions for those users that are going to be using it on a day-to-day basis. And that includes 3-level user access as well. So you can set user IDs as administrators, operators, or supervisors. But you have the ability to log in using the little icon here. So we can select our end user and then input our password. So now we have a specific user logged in for this particular system. And then that will, all of the subsequent runs and results that are associated with this user will then be logged on the system as well. All right. We also have the ability to change our system name if we want to. We're not going to do that right now. And then we just have a regular status indicator here letting us know that the instrument is ready to test. And then once we start running a sample, it'll change from green to yellow, indicating that a test is in progress. Time and date, again, for our compliance folks, focusing on compliance. So all of the results that are logged will have a time and date stamp. Those are all saved on the system. We have our range toggle switch here, so we can again switch between low and high. If we have a rough idea as to what the osmolality of our samples should be, we can toggle it between the low and high sample ranges prior to running the sample. All right. And again, sample ID tags. So again, we have the ability to include sample IDs to run the osmolality test. Right now it's set up to, I don't have it set up right now to run sample IDs, but we have the ability to add them should we want to. You also have the ability to require sample IDs and user IDs for running samples if that's part of your institution's regulation scheme as well. All right, directly below we have our main menu button, which will bring us to our main menu and then our various submenus. We'll dive into that in just a few moments. And then here on the bottom icon, bottom dock here, pardon me, we have a series of icons. So we have our local area network connection indicator. So if we were to network this to our local area network, we would have a status indication here letting us know that the connection was successful or unsuccessful. We have a nice little legend key here. We'll see a few different icons as we navigate through the menus. This will just let us, give us a quick overview as to what those icons indicate. So for our testing icons, we have our sample counter. So the instrument does track how many sample tips are currently left in the consumables box as we do testing throughout the day. That ticker will start from 100 and then slowly work its way down to 0. But once we have less than 20 samples, we would have a visual indication letting us know that it is time to grab an additional box of consumables should we have more than 20 samples to run in this particular testing time period. And local area connectivity indication. So if we have, if it's connected, it'll show with a green status indication. If it's not connected like this one is right now, it's showing as disconnected. Again, we have the ability to enable LIS and LIMS connection as well. And if we were to do that, then we have various indications showing us what successful connections would look like as well. All right. As I mentioned, we have various access levels. So we'll see these keys throughout the submenus as we navigate through them. So red keys indicate only administrators have access to logging into those particular menus. Supervisors and admins are indicated by a single gold key. Operators and administrators with a silver key, and then all users will have access to menus that are denoted with the double gold and silver keys. All right. And then finally, we have our indication icons as well. So we do have the ability to review and sign all results on the system, should we need to do that. And this will give us a nice visual indication as to how many results need review and or approval. And then as well as exporting results, we have the ability to export CSV, results files, as well as PDF files as well. And this will give us a number of files that are due to be exported in the PDF format. All right. And then to the bottom left here, we have our, as I mentioned earlier, our sample tip indicator. So we currently have 77 samples before we need to swap out our XT100 kit. And as I mentioned, this has 100 test sample tips as well as 100 cleaning ones as well. All right, so that's it for the home screen. And what we're going to do next is dive into the main menu and then go through some of the various sub-menus. But before I do that, I do want to just take a quick pause to see if there's any questions that are fluttering through the chats. Okay.
For the low and high range, will the osmometer give an error message if I'm measuring something in the wrong range?
Yep. So the question was, if the sample is run outside of the incorrect range, will the instrument give a warning letting the end user know that the sample is out of range? And absolutely yes, it will. So if, say, we run a high osmolality sample and we run it in the low range, it'll give us a warning letting us know that the specified range is incorrect, and then we'll rerun it with the correct range specified. All right. Any additional other questions? No? All good?
One may come, I'll let you know.
Okay, fantastic. Well, once that does, we'll move into the main menu here. So we have 4 main submenus. We have calibration, results, settings, and diagnostics. We're going to be spending the majority of the time in the results, settings, and diagnostics section for the purpose of today. I will briefly touch on calibration right now. So the instrument itself does not need to be calibrated daily, as I stated earlier in the beginning of the chat. The instrument comes pre-calibrated from the factory, and then it's verified once it's installed in your institution. But the calibration is incredibly stable. You don't need to do it daily, as I mentioned. We really recommend doing it once a year, either on a PM service visit by our field service engineers or members of our tech service team, or if you notice that your control daily verification samples are running outside of spec, then that would be a nice visual indication to potentially recalibrate the instruments. But again, those are really the only 2 major instances where the instrument would need to be recalibrated. We do highly recommend a daily verification prior to running the XT and the OTP. So this can be either with our Clinitrol 290 reference standard or one of our other reference solutions that are closer to the osmolality of your expected sample as well. All right. So next we're going to dive into the settings section, and this is where we're going to, I want to highlight some of the features of the XT and the OTP that really set the instruments apart from some of the other offerings. And again, focusing mainly on the data management, the configuration of the testing and audit trail, as well as the main points that I want to make clear to you guys today. All right. So first thing I want to do is, again, as I mentioned, we have the ability to create unique user IDs. So we can manage the users in this particular section here. So again, this particular menu has a red key indicating admin access only. So I will log in using my credentials. And now we have the ability to create unique user IDs directly on the system. And that's incredibly simple to do. We just hit the add button. We create a unique user ID, a unique password as well. And then we have the ability to select the access level. So again, either operator, supervisor, or administrator. I'm not going to do that right now because it's a demo unit. So we're just going to go back one menu. One other nice feature too is we do have the ability, once the instrument is networked, we can utilize LDAP. So say your institution wants to control the login to your instruments utilizing your institution's credentials. So you'd have the ability to do that once the instrument is networked, and then you can liaise with our tech support team and your IT team to set up the login for use of LDAP. So you don't need to create specific user IDs on the OsmoTECH XT, like I just showed you. So your IT team would have a little bit more control over the access to the systems. Again, if that's something that your institution requires, the XT and the OsmoTECH Pro do have the ability to do that. All right. So next, what I want to touch on is control. So I mentioned this pretty briefly. So we do have a series of reference solutions and verification controls that we can utilize on the OsmoTECH XT and the OsmoTECH Pro. But what's nice is we have the ability to log and track those performance of those controls over time as well. So here we have a list of all the controls that are currently logged into the OsmoTECH XT. So we have our Clinitrol 290, our 500, our 850, and our 2000 controls, as well as our associated launch, expiration dates, nominal values, lower range and upper range of those controls as well. Like I said, so you have the ability to track the performance over time. So if we were to select one of our controls for the example right now, I'm going to do our 290, and I'm going to hit chart. And what this will do is this will pull up a Levy-Jennings plot of our performance of our control over time against varying degrees of standard deviations. So right now we can look at it across, the performance between 1, 2, and 3 standard deviations. And then our data points are indicated by these little magenta icons here. So we can see that we're falling pretty tightly within 1 standard deviation of the specs of our 290 control. And again, so this really comes in handy too if you think that your controls are running a little bit outside of spec. So this is a nice visual indication to track your performance and your controls over time. And then the nice thing about it as well is every time we run our verification, at the beginning, we have the ability to scan in our barcode on the back of our control box, and then the system will recognize that as the particular control of interest, and then it'll save the data and then plot the data in this chart as we go along. All right. And the other nice thing about this as well is we have the ability to create actions for our controls. So say the control that we run at the beginning of our testing day does go outside of our spec range. So we can create an action to issue a warning, and then we can set our range limit for our action as well. So we can set a warning when it's outside 1 standard deviation, 2, or 3 standard deviations. So right now we have it set up to issue a warning if it's outside of 2 standard deviations of the nominal value. So next what I want to do is go into our restrictions. So this is where you guys can get very creative with how you set up your system to be utilized on a day-to-day basis. So again, this requires admin level access for this particular unit. So I'm going to log back in. All right. And then here's where we can define our user access. So you have the ability to toggle between a few different options between your supervisor and your operator level access. So for instance, right now we have it set up where anyone can change the language and the networking, and then the date and time, and then perform calibration are limited only to supervisor and operators. Again, a few other options as well: probe resistance, changing, perform cleaning, hardware options, edit controls information, remove remote lock. Those can all be toggled on and off. Again, this is where you guys would develop your own SOPs and determine which user level access would have permission to do these on a day-to-day basis. All right. You can also customize your passwords as well. And again, you can set the expiration to expire never, or anywhere from 30 to 180 days. You can create your password strength, so specific number of characters, number of lowercase characters, uppercase, numerical digits, special characters, etc. And then testing. So this is where the restrictions really kind of come into play here. So you can set requirements to run a test. So as I mentioned earlier, you can set requirements for user IDs and sample IDs to be entered prior to running a test, and the system will prompt you for both of those prior to performing an osmolality test. Right now we have them toggled off, but again, these can be toggled on if that is part of your testing process. Same thing too for auditing. So as I mentioned earlier, we have the ability to review and approve results directly on the OsmoTECH XT as well as the OsmoTECH Pro. Right now they're toggled off, but you'd have the ability to toggle them on and on and off for the review of results as well as the approval. So right now, if we were to toggle those on, so supervisors and administrators would have the ability to review and approve results. You also have the option to require comments on all your signatures. So once the once the results are reviewed and approved, then the person doing the approval and the reviewing will then be issued to enter a comment as to why they're performing that particular action. And again, these all get saved directly onto the OsmoTECH XT. So all of the data as far as the results, but then also all of the actions performed on the OsmoTECH XT and the OsmoTECH Pro are saved locally to the system. And both systems have functionally unlimited data storage. So for the lifetime of the instruments, the data will be living locally. And then if you wanted to transfer data off of it, it's still going to be lived locally, but we're going to be moving a copy of the data from the XT and the OsmoTECH Pro to either that shared location, your LIMS, your LIS, or any sort of additional locations that you'd want to designate for that data to be moved to. All right. You can also set timeouts for the system itself. So you can set automatic log timeout of 5 minutes, as well as the screen dimming time as well. It's pretty self-explanatory. We're not going to spend too much time there. Data management. So that's going to be our next area of discussion. So again, admin level access. We will type in our password. So right now we have data export enabled. And again, as I mentioned earlier, we have a few different formats that we can export them as. We can do comma separated value formats as well as PDFs as well as the ability to select our location. So right now we have it set up to export to a USB. As I mentioned earlier, we have 2 ports in the back to accommodate a USB drive. Or if we were to network our system, we can select the network location as well as the particular folder that we want to export our data to as well. All right. You also have the ability to back up the database on the OsmoTECH XT as well as the OsmoTECH Pro. And again, the location selection is going to be the same as the data export. So you can either export it directly to a USB or your local area network connection location. You can do it manually, so you can walk up to the instrument and then select a manual backup, or you can enable a synchronous backup, which can take place every couple of minutes, or you can do a scheduled backup if this is enabled as well. So you'd be able to select the cadence, the time of day, as well as the day of the week, and then or the monthly selection as well as to when that database would be backed up. All right. And then on the flip side of that, you also have the ability to import your database back to the OsmoTECH XT or the OsmoTECH Pro, again, either utilizing a USB or the local area network folder to do that as well. And that's going to be a .db file format for both of these. All right. And then with that, I'm going to pause real quick just to see if there's any additional questions in our chat. Perfect time. Fantastic. So the system will retain all of the results and actions performed on the system for the duration of the lifetime of the instrument. So it has functionally unlimited data storage, so it'll take a good amount of years for it to actually fill up. I believe our test team actually kind of did a rough back of the envelope calculation, and it'd be roughly a couple thousand years for it to functionally fill up the database on the system itself. All right, that's it. Fantastic. Alrighty, so with that, what we'll do is we will go back to our main menu, and then we will run a quick sample using our Clinitrol 290. Again, so this is going to be our solution that's going to best represent a solution at the physiological range. So I'm just going to take open our ampule here. Oops, sorry, there we go. Now I'm in frame. Quick crack. So these are going to be our single-use ampules. Grab a sample tip. Pop it on our sampler. Give it a quick. And then now I'm just going to take a lint-free cloth and then just remove any excess liquid that might be attached to the sample tip here. And then we have a slight meniscus, so I'm just going to put that real quick. And remove the cleaning wand from the chamber. Add the sampler to the chamber, and then now I'm just going to plop in a quick sample ID. I'm going to utilize the barcode on our 290 box. And then now our sample is running. So right now what I wanted to do is just briefly touch on what's actually going on here as far as the process goes. So if we kind of look at the front here next to our OsmoTECH XT logo, we actually have an example sample freeze curve. And so what we're going to do, what the instrument's doing is it's supercooling the sample well below its freezing point. So if we were to follow the far left position of the curve here. So right now we're supercooling our sample to a predetermined temperature. And then what we'll do is we'll induce nucleation with a mechanical agitation. So then what will happen, our sample will then release a burst of energy and then go up to its appropriate freezing point. And then that's where it'll plateau. And then the XT is same thing too with the OTP. We'll take a measurement at this plateau, and then it'll convert that to osmolality. And that's what we'll see in about a few moments here. All right. So we see we have a nice little test progress indication as well. And then this mechanical agitation usually takes place right around the 65 to 70% mark. I'm not sure if the microphone will pick this up, but it'll be a series of small strikes that you'll hear as the impactor is going to hit the sample tip on the sampler and then induce that nucleation of the sample. All right. I guess I'll just use this time too while we have a little bit of space. Do we have any additional questions, Eric, in the chat? Okay. All right, all set on questions. Perfect. Oh, there we go. So we've. No worries. Perfect. All right. So right now we had our knock. So our sample is almost about to finish up here. And what will happen here is we'll have our result displayed right above the progress bar. So we have our sample ID, and then once the sample is complete, we'll have our date and time, and then here we have our result. So the system also gives us a nice visual indication to remove our sampler and clean the chamber. So we want to do this pretty quickly because that sample is rapidly warming, and we don't want it to linger on the sample probe too long. So what I'm going to do is I'm going to take our cleaning wand, I'm going to put it directly into the sample chamber. I'm going to give it 5 turns to the left, 5 turns to the right. I'm going to flip it around and then do the same process on the reverse side of the wand as well. All right. So now our chamber is clean. And then this is a nice visual indication too. So I'm going to leave the cleaning wand in the sample chamber. It just lets the next user know that the system has been used, but it's also been cleaned. But then I'm going to put our sampler right back in the cradle. And then what I want to do now is go into our results section. So since we just ran a test, we want to check out the results. So as I mentioned earlier, so functionally we have unlimited storage capacity on the system, and then we have all of our results displayed in a list format. Again, with the most recent sample is going to be at the top, but you have the ability to change the orientation of this as well. So we have our sample IDs, our results, date and time, user ID, and then any specific information related to this sample as well. But the nice thing about this is too, say we want to run our samples in triplicates and we want to do some statistics on this, we have the ability to select multiple samples. And then now we have the ability to run statistics on this as well. So I'm going to use this with one of the other 290s that I ran earlier this morning. So right now it'll display our mean, our standard deviation, as well as our coefficient of variation for the samples that we select. And then this can be exported as well, either to your USB or your network position as well. All right. And again, same thing here too. We have the ability to export. And then if we had our LIS and our printer enabled as well, we'd have the ability to use those functions to export our data as well. All righty. With that, that is everything that I wanted to touch on the XT. And right now I want to move over to the OsmoTECH Pro. But before we do that, I will just ask Eric one more time if we have any additional questions for the XT. A number of questions. Okay, amazing. Yes. So the question was regarding air bubbles in the sample tip. So yes, air bubbles will play a factor in the way that the sample is run on the XT. So we want to make sure that we don't have any air bubbles present in the tip. The same thing will hold true for the sample cups on the OsmoTECH Pro as well. So the air bubbles will definitely impede the way the freeze process that'll happen. So if you do see an air bubble, it just expel your sample and then repull it, and then just make sure that you are not introducing any air bubbles into the system prior to loading it. Yep. And just move over, pardon me.
The result of if the result of the reference standard is outside of the acceptable range, what do you do?
All right, so the question was related to the results falling outside of the acceptable range for the reference standards. So good question. So this could indicate that the instrument needs to be recalibrated, but we always want to run an additional sample just in case to confirm that. And as I mentioned earlier too, we do have the ability to track the controls over time using that control feature that I showed earlier. So you'd have the ability to issue warnings if those controls do fall outside of your specified spec range. But again, not to worry, it's not the end of the world if they do fall outside of the spec range. Again, could just mean that the instrument needs to be recalibrated. But again, the calibration is very stable over time. So there's not really a increased instance where you need to do that.
Does the syringe have to be calibrated or re-verified?
Good question. So the pipette or the sampler, sorry, for the XT was asked regarding calibration on a yearly cadence, I'm assuming with the other pipettes that are in the lab. So the sampler itself is a positive displacement pipette, and it is calibrated whenever we need to change out the plunger wire. So I don't think I mentioned that. So the XT100 kits come with a replacement plunger wire that is set every time we reset the consumables box, pardon me, we want to change out that plunger wire because over time that Teflon tip will start to erode, but we want to make sure that we put a fresh one in every 100 samples. And that's done with a quick calibration. We're essentially just calibrating the depth at which that plunger wire is set inside the sample tip. And it does not need to be included with your regular stock of laboratory pipettes for annual calibration. So the calibration will then fall on the end user for that. Oh, fantastic.
Next one is about.
Yes. So question regarding printers. So we do have a dot matrix printer available that is compatible with both the OsmoTECH XT as well as the OsmoTECH Pro. So if that is something that you guys require as part of your reporting process, we do have options for that. So regarding routine maintenance outside of just the cleaning wand. So we do have a pretty robust maintenance procedure that is detailed very nicely in our manual. So the beyond just the dry cleans. So again, it really kind of depends on what type of samples you guys are doing. If you're doing something that's incredibly complex, as Eric mentioned earlier, so high protein, high sugar, maybe there's some additional cryopreservatives in your formulations. Those would kind of really dictate and drive the maintenance and cleaning cadence of the sample probe. But then not also that, but then the sample chamber itself, as well as the impactor that is creating that nucleation step. So we do have a few other pieces outside of just the dry clean that is looped into our maintenance procedure for the instrument as well. Yeah. Sure.
So is it possible to use a different positive displacement pipette for sample tips?
So good question. So questions regarding using an additional positive displacement pipette with our sample tips. So the answer is no. So the sampler that comes with the OsmoTECH XT is required for use with the XT, as well as the sample tips. So I will just we'll leave it at that.
I'll let you dive into the
Fantastic. Alrighty, so next we have our OsmoTECH Pro. So as Eric mentioned earlier, this is our multi-sample micro-osmometer. It has a 20-position carousel right here up front. So it offers a little bit more walk-away capabilities than the OsmoTECH XT. The range is, again, as Eric indicated, 0 to 2,000 milliosmoles and a testing time of about 90 seconds. All right, and the sample volume is going to be 30 microliters as opposed to the 20 on the OsmoTECH XT. The system itself is self-cleaning, so the end user doesn't need to meticulously clean the probe as well as the chamber after each use, like you do on the XT. So we do have a wiper ring which is attached to the carousel here. So the probe will then lower between different positions on the carousel, and then remove any excess liquids that are potentially present on the probe, piercing the wiper ring, and then removing those prior to going to the next sample, preventing carryover. All right. So similar features on the OsmoTECH Pro as we have on the XT. So we have our integrated barcode scanner right up front here, just like we do on the XT. And then we have the associated ports on the back, our USB ports, our Ethernet port, and as well as our printer port as well. And again, as far as the user experience for the interface, it's going to be very similar look and feel to the OsmoTECH XT. Obviously, the home screen is going to be a little bit different since we are working with 20 samples as opposed to just one. So we have 20 different positions that we could load our samples into. And then when we're ready to run our samples, the system will then scan each available position for the presence of a sample cup. And it'll then log it in the appropriate position. And then again, if we wanted to have this particular function on here as well, we can add our sample IDs as well as our end user IDs to run those tests as well. The consumables are our OsmoTECH Pro 500 sample test kit consumables. So we have 500 sample cups before we need to swap those out. And again, we have a very similar indicator here like we do on the OsmoTECH XT. So we have 465 available tests before we need to swap out an additional consumables box for this. All right. And again, same thing we have on our bottom dock here. We have our same indications for our local area network, and then as well as our LIS if we were to enable on that. We have our legend as well as our general information page as well as our main menu button, and then our status indication here as well. All right. So with that, what we'll do is I'll pop off the carousel, and we'll actually run a sample on this so you can kind of see what the process looks like compared to the OsmoTECH XT. And then we'll dive into the results section on this as well. Again, it's going to be a very similar look and feel to the OsmoTECH XT. Yeah, so we'll just give it a quick pop off. So if you guys were to load, if you had a lot of samples to run through in a given day, you can take the carousel right off and then preload everything directly to the carousel. So what I'll do is I will grab our nipple cup, and then part of my reach, I'm going to scoop back across the camera to get our 290 control. My apologies. Pop our carousel back into place. Add our evaporation lid. And again, since we're working with such a small sample volume here, we want to make sure that the lid is, we want to make sure that we run the especially if we're loading, 10 plus samples at a given time, we want to make sure that that lid is on good and tight. And then we'll hit the refresh button. And then so now what the system will do is, since we took the carousel off, it essentially resets everything. And then now it's going through each available position to identify any cups that are present prior to running the sample. So it'll check all 20 positions. You can see here, I saw one in position number 3. And again, if we had the ability to turn on the sample ID function, it'll prompt us to scan that in or type that in prior to running the sample. Sorry, my fingers are a little wet. Hit play. And then now it's the same process that's going to happen. The sample's going to run. It'll go from ready to testing, which it is, and then in about 90 seconds, we will have our results as well. And again, just to reiterate the testing range here, so we have 0 to 2,000 milliosmoles. That's one range, so it's not split into 2 distinct ranges like the XT is. It's just 0 to 2,000 straight through. All right, and while that's testing, I would love to see if I'll check with Eric real quick to see if we have any questions come in with the OsmoTECH Pro.
So
Okay.
when you stats feature,
Yep.
the results file for the multi-sample analysis appear like a new sample result, or is it stored differently so as not to confuse as a new sample result?
I see. So I think so regarding. So I'm guessing this is in relation to. So the question was regarding running a sample and utilizing statistics, and I'm interpreting this is the same sample run multiple times. So if that's the case, so say we have multiple tubes, say we have one sample in position 1, the same sample in position 2 and 3, we'd have the ability to input the same sample ID 3 times and then run statistics on that particular sample.
But it won't generate any new
Yes.
result in there, so it's not going to confuse.
Yes, and it won't generate a new result. Correct. Correct. All right. All right, so at our telltale knock, test progress is almost done. So if you can see here, we also went from 465 down to 464, and then we had 289 milliosmol result for our 290 control. So right within spec, plus minus 2 milliosmoles for our 290. And right now the system is now running the probe through the wiper ring to remove any additional liquid that might be present on the probe as well. All right. And then I don't know if you guys caught this, but we also had the ability to add additional samples to the carousel while it was running. So there's a little plus icon here that popped up briefly as the sample is running. So if, say we don't load the entire carousel full of samples and we had an additional position available, we can hit that plus icon and then add a new sample to that next available position on the carousel. So it'll still continue to run the sample and its intention, and then it'll give us the option to add the sample at a later point in time. All right. So again, if we go into our results section, so it's going to look exactly the same as our OsmoTECH XT results. So again, we have the ability to select our multiple samples if we wanted to, like the person just asked. And then we can also, again, can run the same statistics as well. Okay. So with that, I think we're had a few minutes. I'd love to field any additional questions for either the OsmoTECH XT or the OsmoTECH Pro that might have fluttered through. The event log. Oh, so let's go through the event log. My apologies. So we go into diagnostics and then go into events. So this is where all of our events and actions are recorded. So this is going to be our complete audit trail for our OsmoTECH Pro, and then the same events log is also the same on the OsmoTECH XT as well. So again, these are all of the actions that are taken by the system as well as the end user. So again, we had a setting change here as our latest event that we recorded. So we removed the review of the results from on to off. And then it records the date and time at which that was done, as well as the user ID that performed it as well. And then these also have the ability to be exported similar to the results. So you can export those directly to your USB that's attached or your network location that you're in your team specify as well. So great questions.
Question, does the Tech Pro allow for user ID and sample ID?
Yes, so the question was related to the Tech Pro and utilizing user ID and sample ID. So absolutely. So that is one of our settings as well, and it's going to be in our restrictions section. So it's going to be very analogous to the OsmoTECH XT. So we can go in and show you how to set that up as well. So in our testing section for required for tests, you can select user ID and sample ID. And both those dialog boxes will prompt prior to running a sample on the system. So again, very similar to the OsmoTECH XT. Same thing will hold true for the OsmoTECH HT as well. And the same thing for the auditing. If we want to set up the review of results and approval, it's going to be the same process on the OsmoTECH Pro as it is on the OsmoTECH XT. Yes.
Are you able to print out this?
Yep. So if we wanted to, so let's go into, so a question related to printing results and statistics. So let's go into our results section, and then I'll grab our last 3 290s that we ran, hit statistics. We can export our statistics dialog box again to our USB. All amazing questions. Oh, okay. Fantastic. And additional questions. Sure. All right.
For the OsmoTECH XT, if we are out of the box with tips,
yes.
on top of the instrument, can we refill it, or do we need a new box?
So regarding the consumables box being depleted of tips, and then the question was relating if we can just refill the box with tips or replacing the box entirely. So the box will have to be replaced entirely. So we do have what we call an assurance tag in each of our XT100 boxes. So that's what allows us to appropriately track the number of sample tips that we've currently used in a given testing process. So we want to ensure that we're putting a fresh box of consumables each time that we need to reset our our 100 test samples.
And that also, you mentioned, that would help prevent.
Yeah. Yeah. So this assurance tag also helps us to prevent the use of other sample tips that are not ones that are produced by us as well. And then we also have the warning relating to the number of sample tips that are getting to the dangerous level as well. So
that same thing.
Yep. Same thing with the OTP. So consumables indicator will flash as well if we're getting close to the low range of our box of 500. So the same thing will hold true. We want to ensure that we replace the box of 500 with a fresh one when we need to swap those out. All fantastic questions. Oh, one more question. Amazing.
Does the OsmoTECH Pro need calibration or only during the daily PM?
Yep. So relating to the OsmoTECH Pro in daily calibration. So the instrument does not need to be daily calibrated. Very similar to the OsmoTECH XT. We, again, really only recommend calibrating once a year. It can either be during an annual PM or service visit, or if you notice that there's some slight drift in the the daily verification that you observe. So again, not required to calibrate daily, but we do, again, recommend that we verify the calibration with one of our standards.
I don't know if you showed the barcode scanner for the OTP. Do you mind just?
Oh yeah. So regarding the barcode scanner for the OsmoTECH Pro, so we do have our barcode scanner directly in front as well. So it's orientated a little bit differently than the OsmoTECH XT. So again, it's right up front here. So if you guys want to utilize a a barcoded sample or utilize the barcodes on the back of our reference standards, you have the ability to do that on the OsmoTECH Pro, like you would on the OsmoTECH XT. All right, one more question. Amazing. Sure. So regarding using Clinitrol 290 versus protanol, so the Clinitrol 290 is great because it's really close to that physiological range of osmolality. Protanol 2 is also, I believe we do have some various ranges for the protanol as well. I know that's more used in our clinical setting, but both are completely acceptable to use. So we highly recommend that you guys utilize a reference standard that is closest to your expected osmolality range. Again, USP does has a really great section as to how to determine the range of that verification standard that you want to use. So I'd recommend referencing USP in that regard. So the question was regarding scanning a box of controls that are expired. So if we go into our settings section and go into controls, again, we're going to use the OsmoTECH Pro as an example here. Oh, fantastic. We don't have any controls here, unfortunately. So the system will not flag the expiration date. So that is up to the end user to do to ensure that the lots that are being currently used are within the shelf life of the product. All right. Yes.
That the XT has issues.
Any matrices that the XT has issues with? Okay, so I'm guessing this is relating to high protein concentrations, high sugar concentrations, and something of that nature. So we'd love to say that we can test everything and all matrices. Obviously, we would love to say that we've optimized for that. But as far as known issues that we've seen, the list is not incredibly exhaustive. I'm sorry, I'm being a little bit vague here. But obviously the super high percentages of cryopreservatives and alcohols and anything volatile will present slight issues. Again, we recommend that you test it thoroughly to ensure that the sample is compatible with the XT. My apologies for not having a completely exhaustive list of known samples that aren't compatible with our systems.
That's one I'm happy to talk about too. You can shoot me an email. I've given my email a few times in the chat, but I'm happy to go through the different matrices and all. Essentially, if it's within range, we are pretty good about doing it. There's very few things that we are unable to test if you're able to pull it up with a positive displacement pipette, and if it's within the range of 0 to 4,000 milliosmoles, we're quite good with it. So. But again, happy to go through it in more detail when we have time, because it looks like we are at the top of the hour. So with that, I will say thank you, Ben. If there's any questions we need to get to, please feel free to add them more in there and we'll address them via email. Thank you, everyone, very much. And I hope you enjoyed this and have a wonderful day. Thanks.